Hello everyone, my name is Osamu Muto. I am a PhD student from Nagoya University and Aichi Cancer Center in Japan. Today, I will talk about our new method, RVINN, for inferring real-time gene regulation dynamics using physics-informed neural networks. As you know, genes are dynamically regulated. One famous example is a phenomenon called transcriptional burst, which refers to rapid temporal changes in transcription. The video you see here is an observation of developmental process in a drosophila embryo. These dynamic GFP signals within the nuclei shows the transcription activity of the even skipped gene, which is important for normal development. This direct observation of transcription is made possible by inserting artificial sequences into the specific DNA regions and then detecting the newly transcribed RNA probes using Web cell imaging. And so recent studies have suggested that these dynamic transcription bursts are dynamically controlled by biomolecular condensates, which includes various transcription factors. And similarly, post-transcriptional regulation, especially RNA degradation, is also dynamically controlled. This study demonstrated global and coordinate changes in transcription and RNA stability after osmotic stress in yeast. In this movie, each dot represents a gene, and the vertical axis shows a log change in transcription rate, and the horizontal axis shows a log change in RNA half-life. To directly observe dynamic RNA turnover, this study used metabolic pulse labeling with nucleic acid analogs. In this method, cells are briefly exposed to using analogs, and then samples are collected at short intervals. This approach allows us to estimate transcription rates and RNA half-lives per labeling period. Okay, so far, these direct measurement methods have described the detailed dynamics of gene regulation. However, these methods require a specialized experiment with additional setup costs. Moreover, there are both pros and cons in this method. Web cell imaging offers high temporal resolution, but genome analysis is challenging. On the other hand, metabolic labeling enables a genome analysis, but its temporal resolution is limited by pulse labeling intervals. Furthermore, nucleic acid analogs are known to have cytotoxic effect, and they can potentially affect messenger RNA processing. So the result might be slightly affected by the labeling procedure itself. Okay, so based on this background, our motivation in the study is to develop a new complementary method for inferring gene regulatory dynamics that achieves both temporal resolution and genome scale. Okay, to explain our approach, let me introduce a perspective from physics. Please imagine that you want to infer hidden dynamics or extract information from small and noisy data. So if you have no prior knowledge about the data, it is difficult to make accurate inferences. However, if you have prior knowledge that there's a governing equation or a mathematical model behind the data, so you can leverage it to make more accurate and interpretable inferences. Okay, I will explain the math model used in this study. So as many of you might already know, we utilize a well-known OD model that describes the mRNA-RNA life cycle. In this model, pre-mRNA is transcribed at the transcription rate alpha and spliced at the splicing rate beta. And then spliced mature mRNA is degraded at degradation rate gamma. This OD model is widely known as the basis of RNA diversity analysis in single-cell transcriptomics. And RNA diversity analysis can recover plausible cell-state transition even from snapshot single-cell data by estimating the ODE parameters for each gene. However, our study focuses on the real-time dynamics. Interestingly, this ODE model was originally proposed by Zaizel in 2011 using the time-course bulk transcriptome data, not single-cell data. So therefore, we applied the same OD framework to model time-course bulk transcriptome data to infer the dynamics of transcriptional and post-transcriptional regulation. Okay, in short, we tackle the challenge of estimating the temporal dynamics of the OD parameters. And actually, there are two existing methods based on this idea. The details of both methods are shown this slide, but here, I just want to emphasize that both methods share the same main limitation. I mean, they rely on analytical approaches. So this means that you need to explicitly choose or fix the functional forms of the parameters and gene expression dynamics before modeling. So because of this, so these methods potentially have limited power to represent complex gene expression dynamics. So therefore, we decided to use a more flexible approach. Okay, we address this challenge using physics informed neural networks, or PIN. So PIN leverages the function approximation capability of neural networks and their end-to-end differentiable framework with automatic differentiation. This approach allows us to keep flexible representation for modeling while ensuring that modeled dynamics follow the solutions of given differential equations. Here is the architecture of our proposed method, RVINN. So RVINN mainly consists of two types of neural network modules. So both modules take time as input. The blue module fits the data at their observed time points. Simultaneously, the orange module estimates the OD parameter alpha, beta, and gamma at arbitrary time points in the same temporal domain. A key feature of our method is that it includes not only a data fitting loss, but also a loss term that evaluates the OD residuals. In addition, we also add a soft constraint on the time derivative of splicing rate beta as an auxiliary loss term. This soft constraint is based on the previous experimental result, and it represents domain knowledge implemented in a natural way. Okay. Here are the results of RVINN's performance on simulated data. As you can see, RVINN can accurately recover and estimate the ground truth dynamics of mRNA and OD parameters, even from sparse and noisy data. For comparison, we also show the result from the existing method, INSPECT. So compared to RVINN, it shows non-smooth and jaggy dynamics with low accuracy. This slide shows a more comprehensive evaluation using different simulation settings with various sampling time points and noise level. In all these sets, RVINN performed well, and actually these results were better than the existing methods. Next, I will show some results using real-world data. So we analyzed two public data sets. The first dataset is time-course RNA sequencing data from MCF7 breast cancer cells treated with estradiol or simply E2. So the second dataset is also from MCF7 cells, but treated with tamoxifen, which is an estrogen receptor inhibitor. So in this study, we analyzed the drug-dependent effects on gene regulation, especially focusing on the transcription repo and buffering. First, I will explain the transcription repo. transcription repo was first reported in this paper published in 2008. Simply put, it is a phenomenon where transcription is also promoted in genomic region near actively transcribed genes. So it's called a transcription repo because it looks like local transcription waves that gradually spread out. So here is an example. As an example, we focused on the ASB13, which is an its responsive gene. So as you can see, the neighboring genes of ASB13 also show activated transcription with a time delay. The animation on the right side shows a cross-correlation matrix of the transcription dynamics among these neighboring genes by shifting a time lag. Okay. And we analyzed the putative molecular mechanisms behind this phenomenon, and then we found a relationship with enhancers. This plot shows the expression levels of enhancer RNAs in MCF7 from Phantom5 data. And as you can see, the expression, enhancer RNA levels are very high in the central region of transcription repo. And based on this, we performed a genome-wide cross-correlation analysis to test whether the distance from active enhancers is related to transcriptional synchronization. Okay, so here is the result. So this box plot summarizes the cross-correlation analysis. So as expected, gene groups closer to enhancer regions significantly tend to show temporarily synchronized transcriptional patterns. Okay, next, we examined drug-dependent and genome-wide changes in transcription dynamics. Due to time constraint, I will skip the details here, but under the estradiol condition, we could observe genome-wide transcriptional synchronization and repo. However, these patterns were not observed in tamoxifen condition. The main message of this slide is that RVINN makes it easier to visualize and evaluate genome-wide transcriptional dynamics with high temporal resolution. Okay, finally, let's discuss the transcriptional buffering. Transcriptional buffering is a form of RNA homeostasis. In short, transcription rates and RNA stabilities adjust in opposite direction, depending on biological condition. This balancing act keeps gene expression stable, even under various perturbations. This phenomenon was first reported in yeast, but recently it has also been reported in disease model, including cancer. And RNA binding proteins, or RBPs, are considered putative regulators for the buffering process. We analyzed drug-dependent dynamic transcriptional buffering. So at first, we defined dynamically buffered genes as those showing strong cross-correlation between transcription and degradation within 1 hour. And then we categorized these dynamically buffered genes into the two groups. I mean, it's condition-specific and tamoxifen condition-specific. And then each group was further divided into the two subgroups. I mean, both transcription and degradation were upregulated or both downregulated. So then we used these subgroup, the buffer genes, for following the enrichment analysis. We performed RVP targets enrichment analysis in the subgroup, the buffer genes. And as expected, RVP targets were significantly enriched among these buffer genes. And for the estradiol condition, CIRBP, which is our RVP, CRBP had the highest odds ratio among all significant RBPs. So this is interesting for us because CIRBP is a known prognostic marker associated with estrogen receptor status in breast cancer patients. And for the tamoxifen condition, we found more enrichment of RBP targets in the subgroup of preferred genes, where both transcription and degradation were downregulated. And these RBPs includes the components of stress granules. Okay, the conclusion of our study is shown here. As future work, we plan to extend our model to infer gene regulatory networks by using differentiable features of neural networks and multiomics data. Okay, finally, I'd like to express my sincere gratitude as a member of the MG Lab. By the way, I missed the group photo due to the proceedings deadline. Obviously, it was my fault. But I also thank the research institutes and funding agency listed here for their support. Thank you very much for your attention.
I had one question about this auxiliary loss.
Okay.
It was sort of like a derivative of beta, and you said it's sometimes known. Could you say a little bit more about that?
Yes, I mean, we can leverage the automatic differentiation. I mean, we can easily obtain the, as you know, neural network is fully differentiable framework. So you can easily obtain the derivative of outputs with respect to input. So in this case, we assume that splicing rate would not dynamically change over short time. So then we add this soft constraint on as an auxiliary loss term. Yeah.
Other questions for the speaker? All right, if not, thank you again.
Thank you.